I'm here with the Autism Research Coalition. Today we're going to be interviewing Dr. Mahmoud Ghannoum. He's the co-founder and chief scientific officer for BiohmHealth. I'm going to give a brief background of some of his achievements. He received his master's in science in medicinal chemistry and his PhD in microbial physiology from the University of Technology in Loughborough, England, an MBA from Weatherhead School of Management at Case Western Reserve University in Cleveland, Ohio. He's published over 350 peer-reviewed publications, six scientific books, and was on the top of 1% of cited scientists worldwide. He's pioneered studies on the fungal communities residing in and outside of our body, coined the term microbiome. And he's a fellow of the Infectious Disease Society of America, past president of the Medical Mycological Society of America, fellow of the American Academy of Microbiology and European Society of Clinical Microbiology and Infectious Diseases. He's an entrepreneur scientist, has launched a number of companies and focused on the treatment of biofilm infections as well as microbial dysbiosis. Dr. Ghannoum, thank you so much for being with us today, and please take it away on your talk on the microbiome and autism.
Well, really, thank you, Alex, for this kind introduction. I appreciate it, and it's my great pleasure to be with Autism Research Coalition. So, as you can see from my title, I would like to talk about the microbiome, gut-brain axis, and autism. I would like also to state my conflict of interest. As Alex said, I'm a co-founder and chief scientific officer of BiohmHealth. Now, the objective of this talk, what I plan to do is just talk to you about the gut-brain axis, how the gut talk to the brain and vice versa, and also talk about how the gut-brain axis influence the neurodegenerative diseases, different types of diseases which in our brain, and the relationship between microbiome and stress. I thought it would be a good idea to give you some idea about how the microbiome affects stress, but at the end, I would like to finish talking about microbiome and autism and provide some insight into microbiome imbalance and how we can address it. So to start with, I want to close this now. To start with the gut-brain axis, what do we mean by the gut-brain axis? As you know, it's basically referring to the communication between the gut and the brain. And the first evidence that there is communication between the gut and the brain came from a surgeon who looked at gastrointestinal juices in people with intragastric fistula. And what they found is that there is intestinal function was related to mood. So if the way your intestine works, it affects your mood. And also, more recent studies are starting to show that our brain talk to our gut, but also our gut talk to our brain. So before we used to think everything comes from the brain, but now we know it's bi-directional communication. It goes both ways, okay? Two-way high street, for example. And this communication could affect the way our brain works as well as how our gut function. And the purpose of this communication, why do they talk together? If you think about it, they talk together to try to maintain homeostasis, which means the harmony in our body, and to protect our body against detrimental effects. Okay? So now the two-way communication between the gut and the brain really occurs through a number of pathways. It's a neural, inflammatory, and hormonal signaling pathway. All of these systems work to connect the brain with our gut. And the gut, in fact, and particularly the microorganisms living inside the gut, really play an important role in processing linked to brain development, physiology, psychology, and behavior. And in the other way, the brain affect the gut as well. Okay, specifically here, the gut microbiome plays critical role in regulation of our mood, anxiety, and pain. Okay, and what's important about this gut-brain axis, it really responds very well to stress, Whenever we have stress, it responds to it. It also affects myelination of the brain, brain chemistry, our immune function, our immune system, and neurotransmission, the way we have hormones coming and talking to us, and neurogenesis. Now, this is a simplified figure of how the two talk together. Okay, you can see in the left hand side messages from the brain. What happens when we have changes in cortisol levels? This could affect the body response to stress and can cause gastrointestinal disturbance. So as you know, when we are stressed out, a lot of the time we have tummy ache, we want to go to the bathroom, and you know why? Because we have imbalance in the gut microbiota. Now, in your right-hand side, when there is dysbiosis or imbalance, dysbiosis of microbiome is called imbalance. When we have imbalance in the gut, what happens? We start to affect the way chemicals are secreted in our brain, and these changes can lead to increased stress, depression, and other neurodegenerative issues. And of course, once we have that, it goes back and affect the microbiome in our gut. So you can see it's a full circle affecting how the gut and brain functions. This is how it is represented. You can see the brain, number one, if we have stress, we could affect the gut microbiome. Also, what type of diet we eat is very, very important, because that also could change the microbiome negatively or positively, depending on what type of food you eat. And once we have this dysbiosis, we can affect, as you know, the gut lining, how it covers our, how it's like in the intestine, we have a tube there, and the tube there, it will allow only good organisms to, good material or good nutrients to pass. But if you have dysbiosis, then we have what we call leaky gut. And in this case, the leaky gut here, things start to go in, toxins, bacteria, other small molecules, which really can affect our immune system as well as the brain. And even our skin could also be affected. That's why we found that people who are taking antibiotics, which kill the microorganisms in the gut, guess what? They start to have more acne, because the microbiota, the organism which lives on the skin, changes. So you can see this is the full circle, how our body react to the situation. Now, how does gut talk to our brain? Really, the major factor in this case is the vagus nerves, which, as you know, cranial nerve connecting the brain to different parts of the body. Okay, and the vagus nerves also affects the central nervous system, effects on our behavior, how we behave, our mood, anxiety. And of course, when we modulate this connection by microbiota, if we affect the brain, as I mentioned in the slides before, by the microbiota, we could affect different things. We could affect fat storage, gut barrier function. Remember, leaky gut, where things can go in and out as they like, can generate low-grade inflammation, can increase our stress, which you expect, as well as increase anxiety and depressive-like behavior. I wrote a paper recently where I described microbiome-based approach to manage depression during COVID. You can see this is all really connected. Now, because we know the vagus nerve is very important in this communication, it really tells you that this could be a good target for us to try to do something or address this imbalance, okay? And when we look, there are studies in animals where showed that if they remove the vagus nerve, they took the vagus nerve out and did what? Guess what? They did not experience any behavioral disturbances. So really, the controlling the activity of the vagus nerves is very, very important. In fact, there are some studies showing that probiotics, such as the strain Lactobacillus rhamnosus, can really regulate our behavior and physical reaction through these vagus nerves. And by doing that, how do they do it? They regulate the gamma aminobutyric acid receptor. Okay, so because of this, we are really in a better position now understanding the basic science of this process so that we are able to translate this into new ways to control the gut-brain axis. Now, what about diseases that are affected, brain affecting disorders that are really related to gastrointestinal manifestations? You can see we list different diseases: Parkinson's, autism, which we are going to talk about, Alzheimer. I'm not going to go over these, but you can see a number of diseases are affected by what happens in our gut. Also, different conditions. We already mentioned depression, anxiety behavior, mood, fatigue, as well as aging. So we have a number of things that could be influenced by the gut microbiome. So now I want to go to talk about two subjects. One is gut-brain axis and the microbiome and stress. And then, as I mentioned before, I'll end up talking about gut-brain axis and autism. So this is the stress and the microbiome. And let's first start by defining stress. As you can see, there are different types of stressors that can be distinguished. We have acute or chronic. Some may occur once while others are repetitive. We individuals have different really perception of the of stress, and some of it is really persistence and with other consequences, or it could be less so. Also, when you get exposed to stressor, it is really well known that we have GI issue, gut gastrointestinal issues. Some even stresses can lead to disabilities worldwide and severe economic burden. Because as you can see, especially with the pandemic, which we just passed through, and we still, I hope, at the very end of it, we had a lot of economic consequences because people are stressed and really suffering. And of course, they could not do their job and they had to go to hospitalization and whatever. So you can see where economic burden is high. So what is the relationship between the microbiome and stress? You know, the first thing is about 40 years ago, it was shown that environmental as well as dietary stress, okay, can affect our gut microbiota in animals where they use mice to do so. Which means if you have stress, what happens, we start to have an increase in the pathogenic bacteria that can cause disease, increased in number, okay? And this is just one example of them. It's called Citrobacter rodentum. And when a person is stressed, it really increases the colonization of this organism, okay? And of course, when you have social stress, what you have increased risk of inflammation-related disease or inflammatory diseases. Why? Because we have an increase in pro-inflammatory genes and expression of them. And as a result, we have an increase in the levels of cytokines, such as interleukin 6, for example, which is known to be a detrimental, it's a pro-inflammatory cytokine. We also know that in acute and repeated stress, the level of intestinal IgA also is affected. And when that changes, it affects our homeostasis, the harmony of our body get disrupted, and we end up having some inflammation. And this has been shown really a lot that this secretory IgA might cause shifts in commensal and possibly results in dysbiosis. In other words, the good bacteria, beneficial bacteria goes down, the bad ones they go up. Now, gut-brain communication during stress. As you know, during stress, we have really changes in the level of central nervous system, different hormones, different neurotransmitters. And by these changes, we could affect the gut neuromotor and secretory functions. We could affect immunity and as well as what microorganisms living in our body. In the other flip side of the coin, when we have dysbiosis, also could contribute to perpetuate inflammation. It keeps inflammation going, further causing a disruption of the gut-brain communication. Some of these effects obviously could be mediated by the host microbe, how you as a human being interact with the microorganisms that live inside you, and the consequences really could occur, and it could be from top to bottom or bottom top to the top. So as we showed in that slide, I showed you the circular one. Okay. So now here is just a slide just to summarize to you the communication between gut and brain during stress and compared to when we are healthy. So here we are in the left-hand side. We have when we are well-being, we are healthy. We have good functioning HPA, which hypothalamus, pituitary, adrenal axis. We have normal behavior. The sensation of pain is really minimal. At the same time, this is at the brain level. At the gut level, we have balanced microbiota. We have, because of this, we have normal gastrointestinal functions. Our immune system is working very well. And also our gut has the right morphology. In other words, we don't have break up in the cell, in the lining of our gut. Because when we have a breakup in the lining of the gut, we have the leaky gut syndrome, and then we have major issues. Now, in the opposite side, the stress at the brain level, we have dysfunction of the HPA axis, we have altered behavior, as well as we have increased pain. In the gut, we have dysbiosis, which could affect neurotransmitters like serotonin, for example, and catecholamine are affected in our gut, and the GABA function. Okay, gut dysfunction, low-grade or overt inflammation, as well as tissue damage. You can see the balance between the two. That's why it's very important for us to try to keep the microbiome in the well-being, not the stress being. So now with that, I would like to go into autism, because I know you all are interested in autism more, but I really wanted just to give you background so that you understand it is really related to many different diseases. Okay, so let's talk about autism. And the audience in this lecture, they know, because of the Autism Research Coalition, they know a lot about autism spectrum disorder. You know, these are a group of neurodevelopmental disorder that really start in early childhood. And the main characteristic of it is that we have problems in communication and social behavior. But there are ASD, there are number of diseases included in it: autism, Asperger syndrome, as well as pervasive developmental disorder, none otherwise specified. It's such a big name. So PDD and OS is much easier. Now, the prevalence of ASD, the autism spectrum, really is changing dramatically. It used to be 4.5 individuals in 10,000 children. That was in 1966. Now, after that, 2006, it was 1 in 110 kids. Now we have 1 kid in 68 children. Was 2010. And I'm sure now we need more data, which is going to show you it's even higher. And what's important about this is that it is not one factor play a role. As I mentioned, there are a number of factors that could affect this and trigger ASD. These triggers are believed to work in concert. They work together, okay? Such that each one, whenever we have one trigger, add another one, we have worse situation and more complications. These are the number of the factors or the risk factors for developing ASD, including environmental factors, genetic factors, and of course, the more recently identified one is the gut microbiota and dietary factors. Okay. Now, what about autism spectrum disorder and microbiota imbalance? Studies, and you can see here there are some references which I put down in all of these slides to support what I'm talking about. We know that children with PDD and OS, and especially those with autism, they show imbalance or dysbiosis in their fecal microbiota. So you take fecal samples, you analyze it, and you find that there is dysbiosis or imbalance. You have more bad guys, less of the good ones. Okay, these studies provided the first evidence that the microbiome in the gut play an important role in ASD. And also GI disturbance, including like what symptoms the kids have: abdominal pain, diarrhea, and bloating, are all really manifested in kids with ASD. Now, the other thing people looked at is what sort of metabolites or small molecules. Basically, these are small chemicals that are produced in the gut. So what they did, people took samples, fecal samples, stool samples in other words, and they looked at it and they found that they really have low levels of what we call short chain fatty acids. These short chain fatty acids, they are normally produced by beneficial organisms, okay, like good bacteria, bifidobacterium, lactobacillus. These, they produce them very nicely, and this, you have low levels of them in autistic kids compared to healthy controls. And really, these what they play a number of roles, which are very important. The primary role for them, for example, is to feed the colonocytes, the cells in the colon. Okay, they are involved in water and electrolyte absorption by these cells as well, by the mucosa, which is covered by the colonic cells. In particular, one of them, of these short chain fatty acids called butyric acid, showed that the really this chemical, it is a short chain fatty acid. It has a number of effects, and these have been demonstrated. Number one, it regulates epithelial transport. It affects positively modulate inflammation. It really reduces the inflammation because and oxidative states become much better. It reinforces the mucosal barrier. Remember, it's very important we have the mucosa intact. It's like you have in the house, you have a tube, we have a pipes in the house. You don't want them to be leaky, because if they leaky, you have all water all over the place. So this butyric acid have been shown to maintain the integrity of the gut lining and basically the tube in our intestine, okay, as well as they modulate visceral sensitivity and motility. Therefore, it's very important to have organisms that can produce short chain fatty acids, because these are very, very beneficial to us. Okay. Now I want to all of this have been an introduction, just to let you understand how the gut work with the brain, how is it related to autism as well. Now, to try to gain more, my son Afif told me, that we really should do a study on the microbiome and autistic kids, because of the way we love kids and we like to support them. And that's exactly what we did. He reached out through Biome and he got people asking them, listen, if you have autistic kids, but what we need to also, one child with autism, if he has a sibling who is not autistic, please send us fecal sample or stool sample. And that's what we did. We had 81 people send us their samples, 51 with autism, 30 they are controlled, but they are within really the same family, which is really very, very important. And these are the demographic description of these, of these patients, whether they are white, Caucasian, what about BMI, all of these we captured them. We also looked at their gastrointestinal symptoms, and we found that, for example, upper gastrointestinal symptoms were higher in cases which are with autism compared to control. Also the same with respect to low GI gastrointestinal symptoms in the lower gastrointestinal tract. We looked at the history if they used proton pump inhibitors, and it's a little bit higher than you have in the control. And we looked at whether they use probiotic or not. So, I don't know what happened here. There is something wrong, unfortunately. I wanted to show you a beautiful video here. All right. So I can't do much about it. No problem. Basically, what we did in that, we looked at the microbiome of both kids with autism, kids without, and when we compared the microbiome, we did what you call clustering. Do they cluster together or they separate from each other? And really, they really separate so beautifully, telling you that the microbiome in autistic kids are different from those of healthy relatives. Okay. This is also, we did additional analysis. We looked at what microorganisms like bacteria that are only present in autism, and what fungi, the microbiome, what fungal community only present in autism, and what present in control. And you can see these are 146 were common between control and autism, 85 control common between control and autism. However, in the control, the bacteria, we had 3 that are specific, only present in autistic kids, and these are the organisms here. And in the fungi, we only have this organism unique to controls, whereas we have 6 fungal members. They are unique to the autistic kids. And why is this important? Because when you look at it, for example, Trachosporum montevidinis, this produces, here we are, Trachosporum montevidinis, you can see it, it's present only in control. So when we look at it, we found that this organism produces potent biosurfactants, which prevents the adhesion or the stickiness of Candida albicans and Candida krusei, 2 different species of Candida, to the buccal epithelial cells, to epithelial cells. So really, it can prevent them from sticking there and causing issues and forming a biofilm. So it's good to have it, okay? And now what we found also, the 2 bacteria, Rhozomonas mucosa and this fungus Thermocytes, they have been associated with infective endocarditis, which really bad. These are bad news, and they really were more present in the autistic kids. When you look at the phylum level, you can see here red autism, blue is the healthy, and we found that in autistic kids we have cyanobacteria. One organism, one bacteria was really highly reduced in autistic kids, and this bacteria is able to break down fibers. So that's why kids sometimes with autism, they don't break fiber very well, okay? When we looked at the bacterial phylum, here we found that there is an increase of protobacteria. This is a red flag for inflammation. It was elevated in these kids with autism compared to the control, and we had a reduction of this organism Bacteroidetes, or the phylum Bacteroidetes, which are a primary producer butyrate, and they are really good guys, okay? Now, when we looked at the Bacteroidetes here, again, you can see how there is a decrease in autism, which is a good organism, a beneficial bacteria. We also saw Akkermansia, which here it really reduced also in autistic kids, which is again a good organism. Rhumnococcus, we found it to be increased in autistic kids, and this is really, this bacteria was found to be present in Crohn's disease patients, where, as you know, they have gastrointestinal issues. Now, when we look at the fungi, that was the bacteria. That's what differentiates us. We look at both bacteria and fungi because they all live together and they interact together. That's why it's important to look at both. And you can see here with the Ascomycota, very high level in the autistic kids compared to the control. And we have particularly the pathogenic organism Candida albicans was elevated, which is a pathogen compared to the healthy. We had less of this organism, of this yeast, and we have a decrease in galactomycins, and this organism is really a beneficial organism. We have it down, okay, where compared to the healthy people. Then we went and did multivariable logistic regression analysis to try to identify, okay, of all of these, what really is very, very significant. And we found 2 things. Number one is this one, which is cyanobacteria. This is the organism where it can break down fiber. We found it to be decreased in the autistic kids, but also we found this bacteria, Delphitia, which increased in abundance, and it's really a pathogen. It can cause form biofilm. You know, biofilm, just to explain to you, it's like the plaque in our teeth. You know, we brush our teeth every day. In our gut, we have also biofilms, and also we found the fish also to play a role in that. So our conclusion of this study is that, you know, we identified bacteria and fungi that are associated with the imbalance in autistic kids. We found factors which are important: male, fish, low fish diet, seizure, and upper GI issue, gastrointestinal issue. So now our goal, we understand now what the dysbiosis, what can we do about it, okay? So our next step was, how can we use all the data we generated to try to understand the mechanism by which these organisms cause issues, and then try to develop a therapeutic or nutritional supplement really to address some of the issues of these kids. Okay. Now I have to be very clear here. If you want to come out with a biotherapeutic, this is like a drug. We have to go through the FDA regulation all the time. You cannot claim nutritional supplements to treat autism, because this should go through a drug. Basically, like you have any antibiotic, any drug, it has to go through a number of phases. So that's why we would like, if we want, there is another approach which is possible, which is medical food, which is not as really complicated and as take forever to do. So there are a number of options where to go from here. But what's really important is, can we identify these organisms that can be employed either as biotherapeutics or, let's say, medical food, okay? What we did, we took 25 different strains in our bank, and we wanted to identify two things. Number one, can we find strains that can break down fiber, okay? And we screened them, and you can see here, probiotic potential is 988. We looked at different fibers, inulin, agave inulin, PFOS, and we looked at the growth rate. Do they grow well? Which means they break them down. And you can see these top ones are the organisms that can do it. The others, they can do it, but not very successful as the top one. The next step is, okay, we took these organisms and we want to see, can they break biofilms? Because we want something that can break down fiber so that the food can be digested better in these kids. But at the same time, we want also those organisms to break down the biofilm caused by the pathogenic organism. Okay, and that's what we found. We identified organisms that can do both. Okay, and here is some of the data. You can, for example, Bifidobacterium brevi, really very effective. This one, as well as this Lactobacillus casei, were really great, and Bifidobacterium longum. So at least this narrowed the number of strains that we should focus on that have the potential or could be a candidate for development of drug for or treatment for autism or the digestive issues. Okay, so we selected these strains that can inhibit the biofilm, that can break down the fiber, and we wanted to study more. Okay, so our next step, that was all in vitro. We wanted to do an in vivo model, and I hope this video will work, where we took mice, we took the organism which caused the bacteria that cause really biofilms, and we fed it to these animals by gavage, and we observed them, and I really hope, here you are, it's unfortunate. I don't know what happened here because I shared it. Yeah, well, sorry, I can't show you, but what this slide shows, it's a video of mice not infected versus those that are infected, and they were able to, what you call, you can see mice that are normal, they behave like normal mice. One of the people who in my group did this study, Janet, and she saw the others that took this organism that caused biofilms in autistic kids, and they were really grooming for, look at the behavior content, they groomed for more than over 3 minutes and was noted on 4 occasions. Usually they groom for a few seconds. They kept doing it for 3 minutes. And here the one mouse was going after his tail around and around and around. So that's why I said, okay, what we really need to do is we need to repeat this study with the autism core, because we are not behavioral scientists. I'm a microbiologist. We know microbiome, but we don't know behavior. So now, as we speak, we are running an experiment with the autism core at Case Western Reserve University to try to see, can we have more quantitative, more solid data sort of from people who know how to do this, and that's what's going on now. So anyway, this is my stuff about autism. Now the last thing, one thing which is very important, how to address it, gut brain access disorders. And it's very simple. The microbiome can be changed, used on diet, lifestyle, and the use of nutritional supplement. Okay, and so I this is how I wanted to understand. It's really a lot of the time it's in our hand how we can do this. I always tell people it's like a garden in your house. You want roses in the spring and summer. Now, of course, it's cold. Now we are in Ohio, so these flowers are coming, going. But in the summer and spring, we need to support them. We fertilize them. We give them the right food, and we try to keep the weeds out. And this is all in your hand. This message is really very, very important. I want to tell you about it. So let me show you here. I don't know what's going on. Okay. These are the diet modifiers. This is the effects of nutrients food on gut microbiota and really pathogenic effects you have if you eat this type of food. Basically, if you follow the westernized diet with all hamburgers, with all fried stuff, with all uh, really saturated fats, you are gonna increase the issues in the brain. Whereas if you change a little bit into plant food, fish, low fat dairy, is gonna help your microbiome to be in balance. Okay, I did a really holistic approach here to try to tell you how we can do that, and I published a book I'll show you later about. It's called Total Gut Balance, which it's published, and we did a study. I will share it with you to show you how following that it will improve the microbiome. This diet is nutritionally balanced, whole food. It has to be rich in fiber and resistant starch because we need to feed the beneficial organisms. Low sugar because fungus, especially Candida, loves sugar. So try to avoid that. Really have low sugar. Now it's full of mono and polyunsaturated fats. Unsaturated fats are better for us, like from fish and this sort of thing, than the others. And also, we wanted to have a diet that's flexible, diverse, and customizable. You cannot stick in a diet which is very, very rigid. Okay, this is this. What our aim was is to have a diet that can limit the growth of pathogenic fungi such as Candida. And of course, studies have shown that if you are deficient in vitamin A, C, and B, you have a really bad situation with respect to Candida. That's why we recommend having these vitamins, low carb, and good balance of plant-based proteins are very, very important. We need to support the beneficial organisms as well, and that's where you eat fibers, okay? And then you break down the biofilm, which I talked about, with vegetables, fruit, garlic, apple cider vinegar, as well as reduce inflammation by eating a lot of antioxidant foods. Very, very critical. I'm not going to go in detail with this. It's all in the book. This is how overview of the balance. It gives you what you use daily, weekly, and this sort of thing. And at the end, there is also a really very good description of recipes, which I worked with a nutritionist and dietitian. She was a pleasure to work with, and we used this diet to cook food, which really was very, very nice. Here is the lifestyle. What we need to do to have a better life is to try to manage your stress, for example, by some yoga, some meditation, walking out. We need to make sure we protect the gut lining. Also, we need to have exercise, for example, activity. You don't have to kill yourself here. Just do 15, 20 minutes a day will be great. Moderate activity, less stress, as well as try to sleep 7 to 8 hours. Sometimes it's difficult to do, but really this is very, very important. And this is the last slide. This is the book which I published. It's called Total Gut Balance. And, these are some of the references I used in my talk. And thank you very much for listening.
Thank you very much. That was extremely interesting, and I hope you have some time for questions and for some discussion here. Um, so it seems that that in terms of what we know, the microbiome, we know so much more about it, the bacterial composition, and kind of what are the species and what is potentially normal. And even there's things that we still don't know. But the fungal community, we certainly seem to know less about in terms of being able to be sequenced and everything and the balance. From your perspective, does it sound like the fungal piece plays as big of a component as the bacterial piece and potentially even more in some of these extreme cases with the biofilm?
I really think this is a very important question. Why? Because as you mentioned rightly, a lot of the people just look at the bacterial community. If you think about it, these bacteria and fungi, they live together. Okay, when they live together, they interact together. And I wrote first an article some time ago, 2016, in fact, and I said it's called the Microbiome, which is the fungal community. That this is an ignored kingdom. It's very important to look at it. And I gave an example where, if you think about it, women, when they take tetracycline, the antibiotic tetracycline, they develop thrush. So what happens? Because you kill the good and bad bacteria, and then you give the opportunity for fungi to grow. So I've been talking about that for years. But the good news is in 2001, the National Institute of Health came out and said, it's very important for us to look at both the bacteria and the fungi, because this will give us total gut community. Okay. And about 3 weeks ago, a paper was published in Cell, which is one of these highly regarded, actually two papers from Israel, where it shows that the fungal community play a role in different cancers as well as different gastrointestinal issues. So to me, I'm very happy because people start to realize that we need to look into this. And also, what's important, you alluded to, is that sometimes it's difficult to identify bacteria, fungi, and analyze it. And that's why my role as director for the Center for Medical Mycology, we study fungus for the last over 40 years. We really optimize the method very nicely how to do that.
Amazing, amazing. So, in terms of, you touched on antibiotics. One of the things from my perspective, um, you know, for instance, if your child is sick and you go to the doctor, it seems to me that there's been an increased willingness to prescribe antibiotics for any kind of sickness. And the thing that's always stood out to me is that there isn't an adopted um, afterthought to, okay, maybe this antibiotic course is necessary, but it should be followed by some kind of, prescriptive probiotic, prebiotic composition. And I think that that's really lacking in the medical community. I'd like to get your thoughts on that.
Sure, I think that's where what we call about antibiotic stewardship. Now you are right. Like you go and you have a little bit of sniffly nose, they give you antibiotics. It could be a virus. This is not gonna. This does not really take care of the virus. What you are doing, you are killing these organisms. That's why we are now really pushing for antibiotic stewardship, because you need to know, as you said, you give it if you have an infection. Don't give it just because, oh, maybe they feel better. Okay. And we as a medical community are pushing for that. In fact, I'm part now of writing a paper on antibiotic stewardship, and there are many others who wrote about that. So it's very, very important, because recently I published a paper where I showed you have two types of probiotics with respect to their activity, some of them narrow spectrum, others that are broad spectrum. And what we showed, the antibiotic that have narrow spectrum, in other words, it only targets few organisms, okay, or one particular organism, let's say staph, aureus, whatever, then it had less activity on the microbiome compared to another antibiotic, which is broad spectrum, which kills everything. So that's why we really need to start rethinking, as you appropriately mentioned.
Amazing. So we have some questions coming in. I'll try to generalize them. There was a question about antifungals, and I'd like to get your perspective on this. You know, would antifungals be helpful with particular fungal overgrowths, or is the biofilm, so inhibitory in kind of preventing anything from getting through? Do you really need kind of a multifaceted approach? What are your thoughts on that?
Yeah, I really, this is very, very, very important. First of all, as I mentioned, I've been working with fungus for about 45 years, and I really participate and publish where I help companies develop their antifungals, and I help all of them. So it's not like I'm biased for one company or another. They come to me, I have a lab where we can do these studies, and we evaluate it. And if it works, we tell them, look, this works. If it doesn't work, we say, sorry, it doesn't work. But anyway, with that background, I really believe the approach which you suggested will be really good. Maybe go first, try to use an antifungal if you think we have candida. And once that is done, you really are advised to use some probiotics, because we need to replace the good organism. The same, of course, applies for antibiotics, because antibiotics kills the bad bacteria. And also we have some good fungus like Saccharomyces boulardii or Saccharomyces cerevisiae, which was used for baking and beer making and this sort of thing. So we need to give these probiotic to try to rebuild it back. We need the good guys back.
Amazing. Now, do you distinguish between, when you say fungi that kind of houses the family of yeasts, but also things like molds, and mold is something that has been talked about a lot, and particularly in autism. How do you distinguish the differences between them, and do you find that mold or types of mold also to be an issue um, in the gut?
In the gut, when we analyze the samples that we receive from people, okay, we really look at what we call all the organisms. When we give the report from Biome, we don't give everything, because there are thousands of, like, honestly, literally, sometimes in one person you may have 1,000 organisms. Okay, so what we did, we select what are the most relevant organisms, and some of them, including Candida, which is yeast, which we talked about, but we also look at Aspergillus. Aspergillus is mold, okay, which is present. So we look at the levels there as well. And now we particularly Aspergillus in the gut is not as detrimental as when they go to the lung. Okay, I've been reviewing the literature recently for the COVID and fungal infections, and really, by far, Aspergillus, which cause pneumonia, is much more dominant in the lung. In the gut, we find it, but we don't see a lot of like sort of out of balance there, because we looked at thousands of people who we classified as healthy controls, and we detected there, but it's not at very high elevated level.
Hmm. Are there any other things that can help to inhibit biofilm? I know that you have this biome test that will show you some, this balance of bacteria and fungi, and you have some products in there that have probiotics. But are there any particular things that help to kind of degrade it in a safe way that you would suggest, either dietary or through an enzyme or something like that?
Yeah, I really think this is very, very also a good question. I think with respect to the biofilms, obviously it's very hard to crack it, okay? But diet, for example, apple cider vinegar have been shown to have activity against biofilms. Garlic have been shown to have activity against biofilms. So some diet will really work very, very nice in it. Of course, with apple cider, you need to dilute it. Don't use it, neat, because it could cause some effect on the teeth, whatever. But these, yes, there are some dietary approaches that will allow or help eradicating the biofilm as well.
Amazing. No, so you were talking before about that video that you had. So just to kind of re-share with the, I know we don't have the video, but we can talk about it. So basically, what you did is you isolated these. Was it either fungi or bacteria that were producing the biofilm, and then you administered them to rats, and you had a control group, and you observed the differences in behaviors between them, and you showed that in the ones that were given this particular, you know, it was taken from an autistic, sample, if you will, that it induced behaviors that could be construed as similar to those of autistic children at
Yeah, this,
times.
yeah. What we did, the experiment, which I couldn't show. Hopefully, in the future we can. Is we took a bacteria, the bacteria that was significantly. I remember we did multivariate analysis, and this was really highly significant. So we used that, and then we what we do usually once you identify it, then we have access to biorepository, which means we have all germs in our banks where we go and get that and then put it into the animal. Okay, put it into the animal and observe the behavior compared to the control. So that's what we did. We take this bacteria, gave it to the animal, and then we observed them. Okay, and that's what we saw. It was a bacteria which, in our in vitro testing, we showed it makes biofilms. So now what we are doing, we are repeating. I told you the experiments with the autism core at Case Western Reserve University. And what we are trying to see is we are repeating the experiment to capture the behavioral aspect, but also I'm working with the gastrointestinal group here where we are looking at the gut cell lining. Is there a, expression of certain genes that are less like pro-inflammatory or less inflammatory? We are looking at the remember the mucosa of the mice and see is it damaged more compared to the healthy control. So these experiments are ongoing. I really hope to have this done within like couple of months. We should have definitely realized what is going on.
Amazing. Are there? I mean, are you getting the appropriate support from research grants and things like that? I know that. I mean, I'll be honest with you. I learned about you maybe about a year ago, but prior to that, I had not been aware of the work that you've been doing in autism. And are there ways that people could reach out to you to help with funding in your foundation, or is that something that's preventing you from doing more research, or how can we learn more about what you're doing and what's the best ways in your company?
I think it will be basically the it would be fantastic if we have help. What we are trying to do, we are a small company, so we are trying to generate this data and then try to apply for an IH grant. The problem with an IH grant, I just for the sake of basically to be transparent, I have a grant from the National Institute of Health. It's called R01, which is one of the big grants, where it looks at the interaction between bacteria and fungi in Crohn's disease. But I don't have such a grant from the autism. Okay, so what I'm hoping is if the company generates this data which we talked about, and then they apply for grant. The problem, this takes forever. You know what I mean? And if we can have any foundation or any support, this will expedite the work we have.
Amazing. Well, thank you so much for all that you've done and contributed, and your research to the fungal mycology and the concept of the microbiome. I think it's only going to improve as we know more and the technology gets better. And I understand your trepidations with trying to get funding and things like that. And I hope we can get some attention there and potentially get some more collaboration with other people, maybe, and maybe move this forward. But this has been really interesting for me. I appreciate your time and your expertise, and thank you for everything that you do.
Thank you very much for having me. I really enjoyed it, and I was looking forward to it. As I said, like I'm excited about this. I know it's, I'm a patient man. I know how things, and I want to move this forward because it's important to help our kids. I really hope. And thank you for having me.
Thank you. Thanks, everyone.
Bye bye.
Take care.